Okay, hello everyone. Let's get started. I'm Brad Weibel, a senior editor at Science Magazine, which is published every week by a nonprofit scientific society, the American Association for the Advancement of Science, based in Washington, D.C. On behalf of everyone on our panel, I want to welcome you and thank you for taking time to join our session. And thank you also for supporting Euroscience, which has put together this wonderful event. These are extraordinary times, we'll all agree, and we can all take comfort and pride in the incredible work that scientists and scientific societies from all disciplines and all corners of the planet are doing to meet the many challenges we face. And you'll see many of these issues and efforts explored and celebrated in other sessions during the ESOF meeting. Now, when we first proposed this panel in July of 2019, we were optimistic that a few people might want to learn a bit about viruses, about what scientists can do with them, and about what they can do in the environment. Fast forward now 14 months, we live in a world that's been transformed, and now even my 4-year-old daughter is asking about viruses in the environment, when she's not busy drawing unicorns. So while we can't say much about unicorns today, we are certainly keen to provide insight on virus research and engineering, applications in the environment, and complex issues that arise. So without further delay, let's get on with our expert discussion of genetically modified transmissible viruses and their intentional release into the environment. So each of our expert speakers will first provide some brief remarks. We will then follow that with some group discussion among the panelists. And finally, then we will leave time to address any questions that arise from you in the audience, which you can submit at any time via the chat function, I suppose. Our first speaker and my co-organizer is Dr. Guy Reeves, an evolutionary biologist from the Max Planck Institute for Evolutionary Biology. Guy will lay out the basic contours for the session, what we're going to talk about, what we're not going to talk about, what's new, what's not new, etc. Guy will be followed by Dr. Ed Rybicki from the University of Cape Town, who is the card-carrying virologist and vaccine biotechnologist on our panel. He will share his thoughts on key technical challenges and opportunities and concerns. Next will be Dr. Philippa Lentzos from King's College London. Philippa is a sociologist and security studies scholar who is closely involved with the Biological Weapons Convention and will share her thoughts from a biosecurity perspective. Then Dr. Pauline Patterson will join us, an expert in public acceptance of vaccines at the London School of Hygiene and Tropical Medicine. She will discuss how this may impact public trust and understanding, issues which are central to her work as co-director of the Vaccine Confidence Project. And rounding out the panel will be Dr. Wayne Sandholz, a political scientist from the University of Southern California. Wayne will look at these issues through the lens of the dynamics and evolution of international norms, institutions, and governance. So please feel free to submit questions at any time, but remember that we will hold off on addressing them until later in the program. And finally, just a blanket disclaimer, the views that you will hear expressed today are our own and do not necessarily reflect the views of our employers and institutions. So that's it from me for now. Thank you and enjoy. And now, Guy Reeves.
Okay, so my job today is quite limited. Just to set things up for the experts. So I want to introduce the key term that we're going to be using, which is the word transmissible, and then give you a little bit of information about the timeframes of the 2 case studies which we're going to focus on. So the word transmissible, we're focusing on genetically modified viruses in the environment that are transmissible. And transmissible means that they can pass from host to host. So in the case of humans, that would be from human to human. And in plants, it would be from plant to plant, or possibly from plant to insect to plant. But basically, this is the way that natural viruses disseminate themselves. They have lots of different names: self-disseminating, self-spreading, and sometimes they're called infectious, but we're going to try and avoid that term because it's a bit ambiguous technically. So all the techniques we're going to talk about, the viruses have been genetically modified to change their properties to do something desirable, and then they're able to intentionally transmit through between hosts. This transmission is not rare; it's an intentional and frequent event. Okay, so the first technique we're going to talk about is transmissible vaccines in humans specifically. First thing I want to make very clear is that in humans, these vaccines do not exist, and there's no clear evidence that anyone is working to develop them. And that's also the opinion of John Hopkins, who wrote on this in 2000, experts who wrote on this in 2018. The idea of, so a transmissible vaccine works differently from conventional vaccines, where you're familiar, where someone asks you, do you want to be vaccinated? And you provide some degree of consent. That's fundamentally changed in transmissible vaccines, where you inject a small percentage of the population and they go home and infect their family and friends. And then as that virus, which elicits the immunity response, spreads through the population, this is in theory a rapid way to community protection. It's actually a very old idea. Technically, it's not that difficult. So there are a few, obviously it raises questions about compulsion, how the virus might evolve once you release it into the environment, and questions about how would our current techniques, regulatory frameworks, show that it's safe and effective. Will they be able to handle this sort of technique? The second, the interest in the, I want to make clear that this, while there's a very strong consensus about vaccines, this particular technique doesn't enjoy that sort of consensus, and it's quite a niche area, though in the last 2 years, there's been an increase in sort of activity, scientific activity surrounding it. The second technique is an agricultural technique, and this is not, and this is a very new idea. It sort of appeared pretty much from nowhere in 2016 as part of a funding opportunity from the US government. And this seeks to provide farmers with the capacity to walk out into fields and say, I would like to genetically manipulate this already growing field using viruses, maybe to make them drought resistant if a drought is coming. And to do this, I will release these viruses, which are likely to be transmissible on insects, which themselves are likely to be genetically modified, and then they will infect the plants. It will transmit across the plants and make the change that I want in days. So again, there are real questions about whether this could be made controllable enough to be useful. Again, it's difficult to see how you can test these things when the first field trial, it might spread outside of the permitted zone, and your first test might be the actual applied release. Now I'm going to give you a quick insight into the two most advanced techniques that of this class. So there's one in vertebrates, which is a transmissible vaccine in rabbits to protect them against the famous myxomatosis virus and the rabbit hemorrhagic diseases, both of which were released intentionally to control rabbits. But Spanish hunters and conservationists like the rabbits, and they want to protect their rabbits from the released viruses. So they generated a transmissible vaccine, which they tested it in the Balearic Islands in 2000. And then in 2002 to 2011, they attempted to get that license. That was ultimately unsuccessful. It's interesting to note that the sort of self-sustaining solution that myxomatosis represents for other people, the Spanish people, is an enduring problem that they would like to fix. Okay, so the next most progressed is the second example in plants. And again, this is from this program called Insect Allies. Oh dear, we've skipped a slide. This is just to give you an idea of what, in theory, should happen next year. 3 groups of contractors are effective to basically, in large, complex greenhouses that approximate the environment, they are supposed to be able to release insects with genetically modified viruses and within a week change at least half of them, potentially gene editing those plants or other kinds of changes. So that's also quite advanced. And then finally, I want to very quickly say what we're not going to talk about. We're not planning on talking about some viral techniques which are intended to stay confined within contained facilities, and examples were spoken about today in the morning in the conference, which is generating vaccines in plants, but also gene editing of somatic cells and then placing them back in the patient. We are looking very specifically at techniques which are intended to be transmissible in the environment. We're not going to revisit the merits of GM crops, or what is the basis of vaccine hesitancy currently, or problems with CRISPR or accidental escape, though clearly they do form the backdrop to a lot of what we're talking about. And that's what we hope is the minimal amount of information you need to be able to follow this. So I will give up this slide share now, Simone.
Super duper. Good job.
Why, thank you. I don't know about the time. Sorry.
Yeah, it hit about 7. I don't know how best to communicate. I tried holding up a sign, but I don't know if you're able to see anyone else, or it's just, I don't know what it looks like from your end.
Brad, you're popping up as little messages. Hi folks, it's a privilege to be here. I'm Ed Rabitsky from the Biofarming Research Unit at the University of Cape Town, and I'll be giving you a bit more details on releasing genome editing or engineered viruses into the environment. I've stolen this slide from Guy Reeves, where the proposition that I'm discussing is that viruses can be released that can manipulate the immune systems of humans, that's person-to-person transmission, or genetically manipulate already planted crops. So potentially insect to plant viral transmission and other techniques. Now, vaccines going viral is something that was actually commented on by a New Scientist in a very recent article, 19th of August. We now have the technology to develop vaccines that spread themselves, which I'm sure also sounded like a really good idea back in the 1940s and 1950s. But there's a very important but. There's a lot of work still to do. Lab and field trials need to check how effective this approach is and look for possible unexpected consequences of self-disseminating vaccines. And if you want an object example of this, you need look no further than the original vaccine. That is Vaccinia virus, which was the virus they used to eradicate smallpox or variola. In Brazil and also in India, the smallpox eradicating Vaccinia virus has become endemic in animals. From the early 1960s, people found Vaccinia virus isolates in wild rodents and sentinel mice, so mice they actually put out to see what they were catching. It was found in non-human primates by 2010. It was found in people and dairy cattle in the 1990s, horses, buffaloes, swine, marsupials, capybaras, etc. It causes sporadic and unexpected and unpredictable outbreaks. It causes overt disease. And the importance of this in both Brazil and in India, where they call it buffalo pox these days, is that there's no clear path to eradicate it. It is now a disease-causing agent that is wild. Immunity to it will also complicate use of modified Vaccinia virus as a vaccine vector for other diseases. And I give you this as an example from July 2018. A vaccine based on modified Vaccinia virus Ankara, which doesn't replicate in humans, expressing Zika virus structural proteins. And if you'll recall, Zika is a problem in Brazil and Central America and in India and neighboring territories. You may not be able to use such a vaccine in these areas without testing people first to see whether they're naturally immune to Vaccinia because of environmental exposure. The other object example, and I think it was probably people in the 50s who thought it was a good idea that people spread polio vaccine between each other. Africa has just been declared free from wild polio. Nature on 28 August points out that vaccine-derived strains remain. And last wild polio was recorded in Africa in 2016, but since August 2019, more than 20 countries worldwide have had cases of vaccine-derived polio. What happens here is oral poliovirus is administered. It does its job, but kids, especially kids, excrete it. It's a fecal oral route virus that gets out into the feces, gets into water, gets into sewage, where you can actually track vaccine where you can track vaccine campaigns by how much live polio virus you find in the sewage. Trouble is, it has this nasty propensity to mutate, and it can mutate back to virulence. And that's what happens in more than 20 countries worldwide, which have had cases of vaccine-derived polio. So there's currently more kids being paralyzed by cases of vaccine-derived polio than the original wild variety. And this as an object example, if anybody ever thinks that putting viruses with CRISPR gene editing technology in them to go wild in the human population, the effects of CRISPR are nothing like as accurate as people claim they were. This is a study from this year where it was shown that CRISPR editing in human embryos showed large DNA deletions, reshuffling of chromosomes, and in other words, completely unexpected and potentially very serious events. Not relevant in humans necessarily, because it's not being punted as something, not actively being punted by experts as something that should happen, but it is being punted for use in plants. This is an object example out of Nature Biotechnology last year. This is a review I wrote of a paper that described transgenic cassava expressing one of the components of the CRISPR-Cas9 system, the enzyme Cas9. And guide RNAs, which is how the technology works. There's a guide RNA which brings the enzyme to the point where it's supposed to work. And that point was going to be infectious African cassava mosaic virus, which is a massive problem all over Africa. And the idea was fantastic, and it worked in cells, and it worked in a few plants in the lab. But when they did the full experiment, it turned out that it probably wasn't viable, because it seemed that giving enough inoculum, which is something you cannot predict, it's up to the insect and how many insects you get on a single plant, completely overwhelmed the innate system in the plant, and in fact looked like it drove accelerated evolution of the Gemini viruses, which are the insect-transmitted plant DNA viruses that cause African cassava mosaic. So a completely unexpected outcome, which was basically an abject failure. This, on the other hand, multiplexed heritable gene editing using RNA viruses and mobile single guide RNAs in nature plants very recently, used tobacco rattle virus, which is a two-component rod-like RNA virus, as a plant gene editor. And what they did is deliver by mechanically transmissible plant virus system to deliver guide RNAs. So in other words, don't make them in the plant, deliver them using a recombinant virus. And Cas9 transgenic plants need to be generated, but you only need to have Cas9 transgenics, and then you could pretty much introduce any recombinant tobacco rattle you wanted to. So in other words, you could repeatedly use the system to create almost unlimited genetic diversity. And because it's mechanically transmissible, it does not have the potential to go wild like African cassava mosaic, for example, would. But the technology would almost certainly not be used in open fields. This is to engineer plants. This is not to engineer them on the fly. For example, you think an aphid-transmitted disease is coming, introduce resistance to it, because the potential that it could get out is there. But the other problem is that RNA viruses are really lousy at being propagated, keeping the trait once they're being propagated. So if you serially transmit the virus, it would almost certainly lose the transgene. But the method could also be used to create very precise DNA sequence changes using other gene editing proteins. So in other words, make transgenic plants with other gene editing proteins and use the same vector to change them. My predictions for this sort of thing are that vaccination by infection of humans and livestock is going to be seen as being potentially dangerous, as it has been for the last 40-odd years, because it's neither contained nor controlled. Dissemination of CRISPR-Cas9 gene editing systems into the wild is most unlikely to happen because of the lack of control and because you're going to lose the trait. Gemini and other plant viruses are too mutation prone to be used for serial transmission of traits. You'd literally have to generate the recombinant virus de novo every time you needed to use it, because otherwise it would simply lose the genes or the RNA. And I'm on such a committee. Any NIH-style institutional biosafety committee would veto any release of technology to permanently alter genomes that was uncontrolled. In other words, via infectious virus or vector, and especially by something like an insect vector. So my final point here is that controlled use of the technology is benign. Uncontrolled is not. And with that, I thank you.
Hello, everyone. I'm Philippa Lentos from King's College London in the United Kingdom. Now, as you've heard, transmissible vaccines face a range of technical challenges, including unexpected outcomes and unintended consequences. But they also raise a whole range of other issues too. So they raise social issues, ethical issues, environmental, political, legal issues. My background is in international security policy, and you'll not be surprised to hear that transmissible vaccines also raise concerns in this area. So the key security concern is really about the dual use potential of transmissible vaccines, as well as other genetically modified viruses for environmental release. In essence, what this means is that the same research that's used to develop transmissible vaccines to treat or prevent disease could also be used to deliberately cause harm. So knowledge that's gained through research on host pathogen interactions or on mechanisms used to overcome the host immune system or the host immune response, or research on viral vaccines could all be exploited to modify immune systems or genomes in a harmful way. So you could, for instance, deliberately engineer triggers into a virus that could cause immune system failures, a bit like HIV does naturally, in the people or animals that it spreads to. Or you could deliberately engineer triggers into a virus that could cause autoimmunity, where the body starts attacking its own healthy cells and tissues. The concern is essentially that while you may intend to make vaccines, others may take your science and develop biological weapons. And the additional concern is that once released, such self-spreading weapons may prove uncontrollable and irreversible. Now, the threat of weaponized biology is not as far-fetched as it might initially sound. We don't actually have to dig very deep for a historical example. The apartheid-era South African biowarfare program clearly shows that intent to misuse biology for harm can arise under certain social, political, and scientific pressures. Codenamed Project Coast, the program was primarily focused on covert assassination weapons for use against individuals that were deemed a threat to the apartheid government. In addition to producing the more traditional injection contraptions that you see on the screen in front of you, it did things like lacing sugar cubes with salmonella and lacing cigarettes with Bacillus anthracis. There have, of course, been many biowarfare programs and several far more elaborate and sophisticated, but the South African program had a project of particular relevance in thinking through the potential dark side to transmissible vaccines. The project was aimed at developing an anti-fertility vaccine. The idea of developing a vaccine that would prevent reproduction took place at a time of general concern about worldwide population explosions. One of the scientists who oversaw the project said he thought the project was in line with the WHO's attempt to curb rising global birth rates, and that it would bring his lab international acclaim and funding. His boss, the director of the biowarfare program, said the military needed an anti-fertility vaccine so that female soldiers would not fall pregnant. Yet in South Africa, this was also a time of growing polarization between the Black and white populations and of increased efforts to preserve white supremacy. And while some of the scientists involved in the project denied awareness of ulterior intentions or even that it was a military project at all, neither of the two chief scientists had any doubt about the real intention behind the project, which was to selectively administer the contraceptive in secret to unwitting Black South African women. In the end, the anti-fertility vaccine was not produced before Project Coast was officially closed down in 1995, 12 years after it was initiated. An early version was tested in baboons, but never in humans. Now, I'm sure you'll agree it doesn't take a massive leap of the imagination to see how the aims of the anti-fertility vaccine project would have benefited from research into transmissible vaccines, particularly if you combine it with current developments in pharmacogenomics. Concerns about dual use are not new. And the good news is that the international community has agreed a treaty completely prohibiting biological weapons. Yet the treaty, negotiated and agreed in the depths of the Cold War, suffers from outdated modes of operation. There are also significant compliance assessment challenges. Now, transmissible vaccines is a small but growing field of interest, as Guy showed you in earlier slides. At the moment, there are roughly only about 10 institutions or so doing significant work in the area. These are primarily located in the United States, and there are also some in Europe and in Australia. But as the field expands, so does the potential for abuse. The field has so far been funded primarily by health and basic science focused funders like the US National Science Foundation, the NIH, the Department of Health and Human Services, and the Gates Foundation, as well as various local institutions. But recently, the military has also started funding the research. Now, military investment for defensive or protective purposes is permissible under the Bioweapons Treaty, but it may still send the wrong signals, and it could cause states to doubt one another's intentions. If one military invests in the area, it is likely so will others. And this then could lead to a form of an arms race in transmissible vaccine work. At a time when the norm against chemical weapons is degrading, you will all be aware of the latest use reported out of Berlin yesterday. We simply cannot afford to have the same thing happen to the norm against the use of biological weapons. It would completely defy the spirit of the Bioweapons Treaty if it seemed like states would even want to pursue the high-risk dual-use activities in biology. Open, multi-stakeholder, and good faith discussions like this plenary about scientific advances that cause particular dual-use concerns are essential to collectively explore the broader stakes of certain technical trajectories. That's all from me for now. Back to you, Brad.
Hi, I'm Pauline Patterson from the Vaccine Confidence Project at the London School of Hygiene and Tropical Medicine, and I'll be discussing public acceptance of transmissible human vaccines and how research about them, as well as the possibility of them being used in the future, may impact on public trust and confidence. WHO refer to vaccine hesitancy as a delay in acceptance or refusal of vaccines despite availability of vaccination services. And what we've seen with the research is that vaccine hesitancy is complex and very context specific. So it varies across time, across place, and across vaccines. And it includes factors such as complacency. So for example, a lack of perceived need or value for a vaccine, convenience, a lack of access to the vaccine, and confidence. So the level of trust, not just in the vaccine, but in the provider and in the process. We conducted a study recently as part of the health protection research in immunisation in partnership with Public Health England, where we asked parents of young children whether they would accept a new coronavirus vaccine should one become publicly available, and 90% responded yes. So for a new vaccine, new technology, this just gives you a bit of example as well of vaccine hesitancy and how people are thinking. So the main reasons that these parents accepted the vaccine was to protect themselves against coronavirus, to protect others, to protect their family, to stay safe for their children, and to stop the need for social distancing. If you look at the reasons why people would not accept or were hesitant to accept, the main reasons were concerns about safety and concerns about effectiveness. And the main reasons for these were that it's a new vaccine and the process is being rushed. These vaccines are being created faster than any previous vaccine. So there are concerns that there won't be enough testing and that it's a new disease. We've only found out about this very recently. So the scientists don't know much about the disease. They don't know much about the vaccine. And there's concerns there that we can think about when applying it to this new technology. If we look globally, you can see there's variation. So here, this is the Wellcome Trust study. It was the world's largest study into how people feel about science and health, with over 140,000 participants in 144 countries. And one of the questions was looking at whether people believe that vaccines are safe. So here you can see, although 79% of people agree vaccines in general are safe, this varies by country, but also it's not just by country, it's by region, by population, by time. How do people perceive risk? So risks that are voluntary, that tend to be perceived as lower risk, whereas risks that are mandatory tend to be perceived as higher risk. And with transmissible human vaccines, people are going to perceive that as mandatory, so as high risk. Also, people perceive risks that are known as lower risk than risks that are unknown. And with transmissible vaccines, it's a new technology, the risks are unknown, and to the general public, they're even more unknown. Something to consider is individual liberties versus safeguarding of the public's health. The role of the state in controlling infectious diseases is widely accepted. It's justified by human rights to health and duty to protect common good. However, the state needs to be mindful of individual rights. Patients want to make their own health decisions. Also, with the internet, with social media, people are more informed and empowered than ever before. However, there's a lot of information on the internet with social media. So there is information overload, there's conflicting information, there's misinformation. And with social media and the internet, concerns can spread quickly and they can spread far. There are ethical issues to consider with self-spreading vaccination as well. The image shows this trolley dilemma. If this train trolley is going to, for example, hit and injure these 5 people, would you be willing to change the direction of the train to hit one person? So saving many to the detriment of a few. So with this self-spreading vaccination, what about those that are vulnerable to live vaccines, such as the immunocompromised? What about their safety? Yes, we might be saving other people, but what about them? And is informed consent possible? Are the public going to be informed? Are they going to be able to decide whether they're going to have this vaccine or not? That would be very difficult to do in this situation. I wanted to give a case study of a dengue vaccine controversy. So in November 2017, in 2016, there was a dengue vaccine that was launched in the Philippines to help prevent dengue and severe dengue. And in November, so the year later, the manufacturer warned that the vaccine can increase the risk of severe dengue in seronegative children. There were signals before, but the vaccine was launched in a country with high prevalence of dengue. So most people had been exposed to dengue. So there would be few people who had not been exposed to dengue before being vaccinated. But if they hadn't been exposed to dengue and they got the vaccine, then the next time they were exposed to dengue, there was more risk of hospitalization. There was huge uproar by the public, as you can imagine, because there was a lot of news in the media that children had died, children were being hospitalized as a result of this vaccine, and the government were accused of committing genocide and having disregard for their citizens. There were insinuations of conspiracy and corruption. Why was this vaccine launched so quickly? There was a change in government, and the new government blamed the old government. So yeah, then there was a huge issue, as you can imagine. The Philippines, they suspended the dengue vaccine program, but we monitored confidence in vaccines in general, and confidence in vaccine safety dropped, and immunization rates as well. So people were vaccinating less. And this is not with dengue vaccine, with just childhood vaccines against tetanus, polio, measles. So vaccination coverage plummeted. There's been outbreaks of measles, and even deworming treatment has decreased. So a huge mistrust in the government, in the public health program. And this highlights how a new vaccine can derail other programs that are going well, other national programs. So it's something to consider when thinking about developing a new vaccine. And my last slide. So with a new vaccine, with a new technology, it will be key to reassure the public with transparency, accountability, timeliness, to listen and engage with stakeholders. So not just to communicate, but to also listen to concerns, acknowledge those concerns and address those, and to reassure the public that any new vaccine will be safe, will be thoroughly tested. Thank you.
Hello, everyone. It's a real pleasure to be part of this panel, and I'm grateful to Euroscience for hosting us, and also to Brad and Guy for organizing the panel. I very much enjoyed the preceding presentations and would like to add some perspective on the potential for and the need for global regulation of emerging technologies, and in particular, of course, genetically modified viruses. It seems highly likely that environmental release of genetically modified viruses will expand across different areas of potential application, but also geographically as more sites and more countries develop capacity to engage in this kind of research and deployment. Now, at this point, experimental releases, and there have been a few, are regulated by relevant national bodies or authorities. The question is national regulation likely to be sufficient? Or is there a need for some sort of global standards on the release of genetically modified viruses? I think the answer is probably yes. And I would like to spend my time today explaining the reasons why I think that's the case. Number one, there are plenty of remaining uncertainties about transmissibility, about reversion, and certainly about public perceptions. And the uncertainties are substantial, which suggests that regulation is likely to vary somewhat across national jurisdictions. That is, the rules will be different in different countries. And that suggests to me the need for some sort of common transnational standards. It also seems to me that the likelihood of cross-border leakage or diffusion is likely to be high. That is, that there are likely to be some sort of international externalities as additional sites and countries engage in this area of technological development. And as Philippa has mentioned, there is a non-zero probability, certainly, of weaponization, and therefore perhaps the need for some sort of prospective arms control or prospective regulation of the dual use technology aspect. Well, colleagues and I have tried to understand the processes involved in the development of international regulation, international rules, and I'd like to just maybe take a brief look at some potential lessons. Oops. For example, if we look at the Space Treaty, this is an example of early international regulation of another emerging technology, which took form in the Outer Space Treaty of 1967. And this, of course, was before space technologies had been deployed on a large scale, which is a bit of a parallel to genetically modified viruses. Of course, the space regime is facing some current challenges, including the proliferation of different private and public actors who are engaging in space activities of various kinds. And again, I think there's a parallel there to the GM viruses. Cybersecurity and cyber warfare is a more contemporary example where there have been some efforts to at least consider global level regulation, but there's been very little progress, mainly because of sovereignty concerns on the part of states that believe very strongly that internet regulation should be at the national level and is therefore a question of sovereignty. So there is the so-called Talon Manual, which was prepared over the last several years, but it mainly emphasizes existing international law relevant to cyber conflict and cyber warfare, rather than developing new norms. Nanotechnology, another current one, again, no global regulation. There's no consensus on whether nano is even a new technology. There are some international regulatory efforts that could be relevant, but they are not obviously relevant to nanotechnology per se. They're more relevant to chemical technologies. There are two international agreements that could be potentially relevant to the genetically modified viruses. And I will mention these briefly. The first is the Convention on Biological Diversity, which is a product of global advocacy culminating in the Earth Summit of 1992. It has very broad membership among countries. And there are some norms that are potentially relevant. For one, the responsibility to ensure that national activities do not cause harm extending beyond national borders. That is the principle of do no harm. There is a duty to assist in technology transfer. There is a duty to participate in the equitable sharing of the benefits, and a responsibility to participate in information sharing. Then finally, the Cartagena Protocol on Biosafety from 2000, it's basically an add-on to the Convention on Biological Diversity. And it does include a provision that's directly relevant to genetically modified viruses, namely the treaty does apply to the transboundary movement, transit, handling, or use of any biological entity capable of transferring or replicating genetic material, including viruses and viroids. And so there would seem to be at least some potentially relevant rules here that could be applied or reapplied or adapted to genetically modified virus. Again, the principle of do no harm, and the idea that there's a duty to notify other countries in case of export or release. There's a duty to conduct adequate risk assessment and a duty to observe an appropriate period of observation prior to deployment. I think there are some challenges that could inhibit the global regulation of genetically modified viruses. Number one, the multiplicity of different kinds of actors who could be involved in this kind of work, not just governments, but private companies and universities. Second, of course, the dual use potential that Philippa has discussed. Third, the technology is still very much future oriented. That is, there hasn't been a motivating disaster. It seems like an unfortunate way to put it, but in fact, there wasn't any progress on ozone depletion until the expansion of the ozone holes was visible and urgent. And then finally, in the current international political climate, there does seem to be a shift away from most forms of multilateralism and international cooperation. Some potentially favorable factors might be that at this point, we're still pre-deployment. There are few releases so far, and in that sense, the space treaties maybe represent an example of global regulation in advance of deployment. Second, there is a small group of forefront states which could make global regulation easier. It's easier to organize cooperation in a small group as opposed to, say, 193 different countries. And then finally, there are these norms and principles, the do no harm principle, precautionary principle, that are in various aspects of international law and could be adapted, I think, to the genetically modified viruses. And that is my last slide. So thank you.
Thanks. Great. Okay. All right. Thank you to all the speakers, and thank you to the audience for paying attention. And please, I just want to remind everyone that we welcome any questions you might have. So figure out how to send them in and get them to us, and we will do our best to address them. Before we dig into some of the questions that we've written down here to discuss, I wanted to first just see if anyone on the panel, we tried our best to keep our initial presentations fairly brief, but now having seen everyone else present, I wanted to give an opportunity if there's anything that you'd like to say now initially to respond to someone else before we move on, or should we just dive into some more structured questions? Any hot takes from anyone here on what you just read or what you just heard?
Could I come in there?
Yeah, sure.
It's interesting, the last talk, especially in terms of regulation and how to govern this kind of thing internationally. Now, there have already been efforts, the so-called Australia group being one of them. But what the problem is, everybody thinks, well, all right-minded people wouldn't possibly do X. And of course, the right-minded people happen to be European Union, the US, Canada, Australia, New Zealand. Turns out that South Africa isn't a signatory to that group. we were prohibited from buying genes, for example, for viruses that are endemic in South Africa that we were trying to make vaccines for, because that was a way of controlling proliferation of potentially dual-use agents, which was ridiculous beyond belief, because in order to do it, we would have had to get permission from the German federal government and then wait 6 months to get anything. So we went to colleagues in South Africa, got the native gene and used that. Now, that's the point. In places where something is endemic, banning people from working on those very things is short-sighted, elitist, and very, very discriminatory.
Point taken. Anyone else?
But that's not a dig at Wayne.
Yeah, not at all, not at all. I think one thing that I don't want to speak for the group, but I think I speak for the group in that we didn't intend for you to audience members to walk away from this saying, oh, I've just been preached at and told.
Brad went dead.
The radar and raise awareness. Hello?
You went dead there for a while.
Oh, okay. Sorry. Can you hear me?
Yep.
Okay, sorry. So yeah, I guess my question is, some would argue that just even talking about some sort of controversial, potentially dangerous things like this might create an information hazard. Others might suggest that it creates some potential, alarmism or misunderstanding the public. Suffice to say, how do you balance talking about things and having transparency and public awareness with talking about it in the right way, in a responsible way? I'd like to hope that we're doing this, but there are many people here who are much more expert in what are the responsible ways for discussing these issues, and what, if nothing else, what should we be working on to say we need to talk about this, and we need to talk about them in the following ways so that it's done responsibly. So Pauline, Philippa, Guy, Wayne, all of you, I'm sure, have experience and thoughts here.
Maybe I'll start then. So I think, I'm really keen to hear what the audience are thinking as well, and I'd be really interested in your concerns and questions. So do write in the chat box, do send us your questions now or in the future as well. And I think there is this issue that you don't want to start asking people how they feel without them knowing about it more, because for someone who doesn't know about this, they might have a red flag alert. Wow, is this happening? And just fear will kick in. And so then there can be concerns. So we want to be careful that we're not creating concerns before they're needed, I guess. But questions are healthy. But we do need transparency, and we need to make sure that governments and institutions aren't hiding things from us, from the public. Otherwise, there will be distrust.
Yeah, I'll come in and just echo what you're saying, Pauline. Essentially, I think from researchers to public, there's absolutely this need to be transparent about the sort of work that's going on, and to talk honestly about not just the potential benefits, but also the potential risks. So we as a community can have a discussion about that. And that also is echoed at the international level, where different countries also need to be open and transparent about the sorts of work that they are carrying out, and particularly about the justifications of the sort of work that they're carrying out, so that people can and states can assess what they think the intentions are. Because if you say there's secrecy, or if you say there's obscurity of intention, that's when the mistrust starts, really. So I think what you're seeing in terms of a debate between, say, the research community and the general public can also be echoed at the more international level, between states and between different institutions.
Yeah, if I might, I would add to what both Pauline and Philip have said, that the multidisciplinary nature of this panel, I think, is a good step, That is that these problems have to be looked at from every dimension, not just the technological and scientific, and of course the public perceptions and dual use and regulation, all of that, but even also ethicists, to bring in ethicists who have the, whose job is to think about the moral and ethical implications of these kinds of developments. So there needs to be cross-national openness and transparency, but also every perspective probably needs to be heard.
And sorry, if I could just jump back again, and I won't hog the floor too much, but in preparation for this panel, I read a lot of the scientific articles on transmissible vaccines to get up to speed about what the science is about. And really the only challenges that were listed were technical challenges. There were really no consideration of broader social, political, ethical, legal questions. And so actually I have a question back to you, Brad.
And I think in my mind, at least, this ties in.
So my question back to you, Brad. So it was really, we lost you there for a second, is really what is the role of journal article in pushing other perspectives and of journal article reviewers in asking for other perspectives to be involved in articles that are looking at these very technical issues?
Ooh, now this is where I speak for the entire, academic journal publishing community right now. From my experience, I make it a point to, when I get technical papers like this, I certainly am obligated to approach a technical reviewer, but I would never comfortably go forward unless I did talk to someone, a social scientist who studies ethics and governance and public deliberation, or a legal scholar or an economist. And so, for example, the paper we published by Guy several years ago talking about this, absolutely the people that I talked to had expertise in not necessarily the lab bench, but broader, how do we govern this as a society responsibly? If I might now, unless anyone else had something, I think one of the the only reason I think we would even be having this discussion is if the powers that be said, well, you know what, we have these problems that we face as society, and the way we've been trying to address them hasn't been working. Our vaccine efforts haven't been working well enough. Our agriculture hasn't been working enough. And clearly, whatever problems we face, this is the solution to it. And I'm not sure that I understand how clearly and precisely has it been articulated. Here is why the solution to the problems we have is this technology. So having said that, what do you all know in terms of how have these technologies been motivated? Has it been a really well-articulated problem that says, oh, clearly this is the solution? So that's open to anyone. Don't all jump in at once.
I would say, I will answer that question. I think there are limits on the value of transparency, and it relates to what Brad just said. You not only have to be sort of transparent and friendly, you actually need to be plausible. Where we are having these discussion about these two techniques, both of which have received or are receiving tens of millions of dollars in research, yet we're still sort of in the dark of what is the justification. I mean, can they get us to an endpoint we couldn't get to with conventional vaccines, or is it just an efficiency argument? And the bit for me that seems to be the most sort of insightful point that I think I'm really quite frustrated by is the upside is always great. I mean, it's very easy to make the upside, but isn't there an obligation, a moral or ethical obligation, to have a plausible path to the upside? Because if you don't have that path, then all you've got is the downside, the interactions with vaccine hesitancy, or potentially start eliciting an arms race, buying into a situation that doesn't really exist. So I think it's true to say that all vaccines will need to pass through phase 3 trials. Should not, before you get funding, you should explain what a phase 3 trial of a transmissible vaccine might look like. If for agricultural solutions with insects flying around with transmissible vaccines, should you not explain how we can establish that they're safe and controllable? Because if you can't establish they're safe and controllable, what have we really got?
Can I come in there? You know, we're now in my province, the sorts of things that we actually try and do. And my wife, for example, makes recombinant pox viruses as animal vaccine vectors, but they're not transmissible, as in they do not easily go from animal to animal. And that is the point, because there's more than enough stuff out there. An example here is African horse sickness, which is a major, major problem, which has just been, it managed to export itself to Malaysia and Thailand, where they've never seen it before. You know, it kills horses to about 80% efficiency. It's a live vaccine, and the thing recombines and reassorts with wild type viruses. And okay, Africa is the only place that has the wild type viruses. But the problem of using the vaccines outside of the country means that you now have a live infectious agent if you have the midges that can transmit it. And all of a sudden, the law of unintended consequences kicks in, and a virus that you didn't know about that infects some local antelope suddenly reassorts, and you end up with a vicious pathogen that starts killing cows. Literally, this sort of thing could happen. So having wild infectious agents out there strikes me as a virologist and a biotechnologist as being utterly crazy. Now, to get something out there that is completely innocuous, well, the common cold is completely innocuous, but the odds are that a couple of the viruses that caused it looked like the coronavirus that's causing the current pandemic back when they started. So in other words, much higher kill rate. But at the moment that the entire population has been exposed and you have background immunity, that things cease to be a problem, but then it's a circulating virus. It's not a vaccine anymore. Lack of control in this sphere is absolutely crazy because, as I said, the law of unintended consequences kicks in, and every good zombie story starts with a virus that escaped from a lab.
Zombie stories keep me awake at night. Anyone else? Philippa, Pauline, Wayne, want to chime in?
Yeah, I'd like to come in on this, because really if you're going to judge intent, you're going to need some details, right? And as Guy was saying, the aims, the expressed aims of some of these projects are incredibly broad. So the one slide I showed from the DARPA program, one of the aim of the entire DARPA program there is really to protect US military service members from spillovers. Now, that's a great aim, but there's a lot of steps to get to that aim, right? And so I think what you'd like to see, what you'd need for true transparency are more details on the sort of scientific steps you need to take to get to there, but you also need, you'd want to see the logistical steps you need to take to get there, the regulatory steps you need to take to get there. And it's similar with the projects that you see in the agricultural field. Again, there's, for instance, another military-supported project there where the aim is merely to protect crops and ensure food security. And again, there are very few details provided in how you actually get to that point. And so it's really just to second what Guy was saying in terms of the need for convincing aims and detailed steps of how you actually get there in order to provide that sort of transparency into your intentions.
Excellent points. Any other thoughts? I guess I would get in over my head here. And if I am playing devil's advocate and I'm looking back at how historically technologies evolve and regulatory regimes evolve. It is not the case that we only proceed to research a topic unless we know in advance how the regulatory scheme is going to play out. So that's just not how it works. So, one could argue, well, this is just an example of that again. Like, we do our best and we go cautiously and we just make sure that we're talking to the regulators and we're doing it safely. And we have hope that eventually we'll get to a safe solution. And so let's give benefit of the doubt. What would that have to look like? What are the steps to help us get there? Maybe we already discussed it. Maybe it is just public needs to be aware. Maybe there needs to be more transparency. But do we have any roadmaps for what it looks like to get there? Anyone?
If I might, that seemed to me to be the crux of Guy's question, that there might not be, or at least it's not immediately evident that there is a pathway to the imagined positive uses of this kind of technology. And if not, then we're only left with the downsides. So there are enough, I think, basic principles of regulation that should lead us toward an extremely cautious approach. Namely, the potential for catastrophe is quite high, because once these things are released, they might not be containable, they might not be retrievable. They can obviously mutate and revert, which would suggest that the approach should be almost a prohibition until there's proof positive that release can be managed in a way that's safe. And I guess I lean toward the Guy's skepticism that there might not be a path to the upside.
Thank you.
I have a question for Pauline. One possible view of people's nervousness about the speed at which the COVID-19 vaccines, the Project Warp Speed, I'm sure no one would express it this way, but one way of saying it is people find the conventional phase 3 trials to be extremely important to them. And when they're compressed or the discipline is sort of changed, this has a lot of knock-on effects. So whatever we propose for these techniques, the phase 3 trials, the mechanism needs to continue to exist for all the other conventional vaccines simultaneously. If any new avant-garde technique comes and knocks out the regulatory system, I know regulation doesn't have a lot of fans, but it actually is really important. I don't know, could you perceive the testing of these techniques, just speaking what Wayne just said, is a potential threat to the regulatory systems that we all rely on?
Thank you. I think it's really key to think that, what I wanted to add anyway, is that even if things don't go wrong, even if things go well, even if there aren't any issues and everything goes swimmingly, misconceptions are going to arise, misinformation will spread, and people may have concerns that they will share with others, and there will be some people that will spread concerns on purpose. So, for example, the coronavirus vaccine trial in Oxford, the first patient, there was misinformation spread that that person had died. That person did not die, but someone spread that misinformation. So this is key to think about, and trust takes a long time to build and seconds to lose. And with other research looking at misinformation and misconceptions, scientific psychological research shows it's really difficult to change people's misperceptions. Even discussing a myth, like I was worried even talking about that some people may come out with a high risk perception, even when discussing myths and misperceptions and concerns that aren't real. So this all needs to be really, I'm not sure if I answered your question, Kai, but this all really needs to be thought about, and before it's too late as well, because people will, even in the development of this, there can be issues with public trust, with public concerns, with misinformation. So this needs to be thought about now, not tomorrow, not next week, not next year.
Thank you. I believe we have received a question from the audience that if Wayne feels able to address it now, would you like to do that, Wayne?
Sure. The question has to do with the Cartagena Protocol and what is missing. That is, I suggested that in principle, well, explicitly, it should cover genetically modified viruses. So the question is, what's missing? What would need to be done? And the questioner actually, I think, hit on the major point, which is mechanisms or processes for risk assessment. That when the Cartagena Protocol was negotiated, I don't think the environmental release of genetically modified viruses was yet very present in anybody's mind. So the treaty would have to be adapted to a technology that wasn't contemplated at the time the treaty was designed, and that would mean I would think spelling out some protocols for risk assessment, for containment, for the sharing or transport or movement of the genetic materials, and so on. And that would have to be done in some sort of expert group, which could then develop a set of standards and protocols.
Thank you. Ed, did you want to weigh in on this as well?
It's a very good entry point, because my wife actually works with a bunch of people called the Brighton Collaboration who look at doing exactly that. That's developing risk profiling algorithms for recombinant viruses or vectors. They've got a publication that's just about to come out on modified Vaccinia ankara. So in other words, the engineered smallpox vaccine. So these tools are there. There are groups that are doing this sort of thing. Whether they get to the national attention, the level of national attention, I'm not absolutely sure. But some very prominent vaccinologists and virologists involved in exactly this. So there is experience, there are materials, and there's a lot of information.
I think I would also just come in on this and say, from what I've understood from the people who are doing this sort of work, they do see that there is a pathway, in the sense that you first do the modeling, you then do contained testing, then you take it out to limited field trials on an island, for instance, then you put in technical measures to make sure you only hit one species and not others. It's, so I think they see that there is a similar sort of trial path as you would have with your trial phase 1, 2, 3, 4 guy. But what I think is missing here is that you can't just have a technical fix to this path. There has got to be broader consideration of this impacting society, different people, different kinds of people, different kinds of countries, different regulatory systems. So I essentially agree with you. I just think that there is a sense, some people certainly have a sense that there is this path, but I think I would certainly argue that you'd need a larger, not just a technical way forward. You'd need much broader considerations as part of that pathway.
I wonder what everyone would think to this. I wonder if there's a sense that our regulatory sort of horizon scanning approaches are looking for new technical developments, which then lead to concerning situations. But as Ed mentioned, particularly with vaccines, these are not new ideas. These are old ideas, and there appeared to be an evidence-based norm that they were not a good idea. And that norm there's an argument that this norm is now breaking down. And this is exactly the sort of situation you almost have to face backwards to notice this one, and whether we are set up to do that.
It's quite disturbing to me that if once you get old enough, you're remembering things that other people have never read. And that this is a fantastic idea. And he said, well, someone's doing that in the 1970s. I'm absolutely positive of it. In other words, if it isn't digital, a lot of people have never seen this kind of material. So I think it's incumbent on groups like this to actually make people aware that there is a body of information out there.
Anyone else want to weigh in on that? That was a question that I wanted to ask Guy, sort of a two-faced question. One was, we're reminded every day that there's the world that is governed by clear codes and rules, and then there's the vast majority of society that is regulated by norms of behavior. And when we see people blowing by well-established norms, you're like, wait, you can't do that, but I guess you just did. And so what was it that was the underpinning for these norms, as you described, that, yeah, we know this is potential, but it's probably not a good idea to do it? Is it simply the fact, as Ed maybe suggested, that maybe people just haven't read the old literature? Or is there a case to be made that, oh, well, any discussion of the norms has to say, like, what's the upside, what's the downside? And, we believe that technology has gotten us to a point where we can be more accurate and precise with things now than we could 40 years ago, or the risk environment has drastically changed and therefore there's more benefit. I don't know. I'm just, to the extent that any of you guys might speculate, has there been, if we want to be generous and say, oh yeah, certain key things have changed that maybe I could see why people would question this norm, what might those things be? Is it purely just a technological, we have more technology silver bullets now, or what? Anyone?
Yeah. I think we've got more different kinds of bullets. Sorry, Wayne.
Go ahead, Ed.
We have more and different kinds of bullets that doesn't necessarily mean you know where they go. That's the problem. Exactly. The moment you expand your arsenal, you have absolutely no idea what some of the effects are going to be, and there will be unintended consequences.
Yeah, I completely agree. And I think that maybe today, more than ever, we do have a sort of technological or scientific optimism, well, which is founded on the many scientific and technological successes that we've seen the benefits of. But on the other hand, maybe it does blind us in some sense to a proper appreciation of the risks, and in particular in an emerging technology where there is so much that we don't know, and we don't really know what we don't know, in which case our innate technological optimism leads us to go too quickly down paths that have not been properly scouted out.
I agree with you, Ed, in that we do need to know our history. We need to know what's been written before and what the arguments were before. But it's also important to take contemporary contexts into account, because I do think contexts change. So to answer your question, Brad, I think what we need to do is to more explicitly articulate the contexts under which we're willing to take on the higher risks that would be involved with releasing these sorts of viruses into the environment. Because perhaps there are very specific, very narrow contexts in which we would be willing to take on higher risks than in other contexts. And to start having this discussion about a more nuanced discussion about the sorts of contexts where we would clearly not take on these sorts of risks, and then move towards the sorts of contexts where we might be willing to do so, and then also find that gray area where there will be very different opinions depending on who you ask.
I want to jump in as well, if there's time, to say we've seen with the coronavirus pandemic that viruses don't respect borders, and how quickly the virus has jumped with globalization, with people hopping on planes. Will this transmissible vaccine in the future become a circulating virus, and do we know the answer to that?
I would say that there is, so I will just answer this very quickly. The Spanish rabbit example actually does provide some insight into that. So the Australians were very happy about their myxomatosis and all the dead rabbits, and they were furious at the Spanish for doing the field trials. They had calculated that within a year, their favorite tool would be compromised. So one person's, I mean, it does come down to values. One person's self-sustaining solution could be another one person group's enduring problem. And I think very quickly with viruses, you may end up on a treadmill, which might just start with the first trial, because most of our regulatory systems do not consider the possibility that when you end the phase 3 trial or you end the agricultural field trial, that the trial then ends. With the transmissible solutions, it may just keep spreading.
Thank you. We have just a few minutes left. Maybe this is too big of a question, but I'll throw it out there anyway. Wayne, you mentioned how, for better or worse, we're at a situation that it might be beneficial that there's only a few small number of countries that are really doing this, and that can help in terms of how we approach regulation. But the flip side of that is that everyone else has a stake in this too. And so the institutions that we have out there in the norms, do we have a way to make sure that this is not just something that is decided by the US and the EU and China and Australia? Like, is the Bioweapons Convention, is it the CBD? Do we, I mean, these all have strong international representation, but you know, are they ready? Are they built for this, you think? Those are the tools we have, and stick to them?
Well, they would clearly have to be developed in new ways and elaborated in ways that would be appropriate to this emerging technology. But I think the raw materials are there. That is, the basic principles and the general norms that govern biosafety with respect to genetically modified materials are there. The one advantage of having a small group of leading states is that it's easier to get coordination in a small group. And then if that small group can produce standards that are so, shall we say, self-evidently appropriate, then the hope would be that new entrants into this domain would accept them and adhere to them.
Thanks. Oh, go ahead, Philippa. Go ahead.
I would just second what you're saying and really say, you know, I think the institutions and the countries that are doing this sort of work have an added responsibility to take a moral lead on what they are doing. And grouping together like-minded states is certainly a good idea to set the bar high to start off with. But it's really the duty of care is really on the researchers who are doing this work to start off with.
Great. You know, we have a minute and a half left, and so I want to be sensitive to everyone's time. I want to thank all the speakers. I want to thank the meeting organizers for making this happen. I'm going to go out on a limb and invite any audience members who might have any other questions to email people here and bombard us with questions just to help keep the discussion going and tell your friends. But you know, 1 minute left. If any final parting shots, guys, now's your chance. So otherwise, we'll just thank the audience, and I'd like to definitely thank the speakers for dealing with me and putting up with me all these months. And good luck, everyone, and enjoy the rest of the conference. And I guess we'll sign off for now.
Brad, thank you.
Thank you.
Thanks a lot. Okay, bye-bye.